Hi everyone, my name is Mara Burrows. Today is March 28, 2020, and this is for my Bio 225 class, week 3, lab 5. Today I am doing exercise 1, which is direct and negative stains. So I already have all my protective equipment on. I've already disinfected my workspace with bleach. Next, what we're gonna go ahead and do is take a slide like this, and we're gonna label that slide 1-direct-M, which I've already done on this slide right over here. Okay, so the M, it just indicates that the sample is gonna be taken from our mouth. So what I'm also gonna do on here is I'm gonna go ahead and just draw a small dime-sized circle just underneath the slide. Okay, done that already. There's that. Next, I am gonna take the sterile swab and I'm gonna scrape the inside of my teeth and gums with it. Just one. Just have to vigorously do it, so I hope. So. Okay, next, it's asking me to go ahead and take this swab. It says set the slide so the label is legible facing upward with the drawn circle facing downwards. Okay. Place the swab only inside the area of the drawn circle and apply the smear to the slide using a circular motion as shown in figure 12. So, I'm gonna take this and I'm just gonna swab the inside of the circle. Okay, I have done that. I'm gonna just go ahead and let this completely air dry. After that, while it's drying, I'm gonna go ahead and light a tea candle up carefully with a lighter. Okay, so now I am gonna go ahead and light this tea candle. Tea candle's on. Wow. And I'm gonna orient the slide so that the smear and the label faces upwards, such as this, and I'm gonna go ahead and hold the slide with the test tube clamp and put it over the flame 5 times. No, I'm gonna improvise. I'm gonna go ahead and use my hand 5 times. And we'll just hold it over where it's smeared on. So 1, 2, 3, 4, 5. Okay? Next, I'm gonna set that aside and I'm gonna go ahead and let that cool. For about 10 minutes. Okay, so it's been about 10 minutes since we let the slide cool. What I'm gonna do is I'm gonna place this slide in the smearing tray with the side up, like this. And I'm gonna locate the crystal violet gram stain, and I'm gonna add 1 drop to the center of the circle. 1 drop covers the entire circle. So what we'll do is we'll let that sit for about a minute. Okay, so it has been staining for about a minute. What I'm gonna go ahead and do is I'm gonna go ahead and take this off by letting it, I have some tap water on here. So I'm gonna go ahead and just kind of rinse it off very carefully so it kind of goes clear until the water runs clear. Okay, that looks about clear to me. So next I'm gonna go ahead and kind of just take off the excess water around it. I'm not smearing, just blotting to ensure that it comes off. Okay, so this is what our stained slide looks like. Just gonna blot it so that it doesn't come off. Okay, so that's the stained slide. Next, I'm gonna go ahead and take a picture of it, and then we will put it under the microscope and take a look at it at 3 different magnifications, and I will go ahead and post it to see which one looks the best. Okay, so we finished with the first slide, and it was actually pretty cool to see. They were all stained purple. Now we're gonna grab a second slide, and we're gonna label it to Neg M, which I've already done over here. Next, I have my Congo red stain, which is this, and I'm gonna go ahead and use a wet paper towel to make sure it's dried off after I cut it. Okay, so I went ahead and cut my Congo red. I'm gonna go ahead and place it on this slide over here. Okay, so I'm gonna go ahead and place just 1 drop on the slide just inside of the labeled edge. So 1 drop, okay. Next, while that's there, I'm gonna go ahead and grab my second sterile swab, and I'm gonna go ahead and scrape my teeth and gums with it again. It says vigorously, so.
Why do you?
Alright. Okay. So after doing that, I'm gonna go ahead and place the swab in the drop of the Congo red stain, and I'm gonna agitate the sample to transfer the cells from the swab to the stain. So. Okay, so after doing that, I'm gonna go ahead and grab another second blank slide, and I'm gonna slide it across the slide. Like so. We're gonna go ahead and let it completely air dry. Okay, so I've went ahead and let the red Congo slide dry off. There are a few spots on here, so next I will go ahead and put it in the microscope to take a look at it at all 3 different magnifications, low, medium, and high, and I'll go ahead and pick which one I feel it looks better under, and I will go ahead and post that on my lab notes.